Preeti Krishnamani. She's a senior at the Charter School of Wilmington, and she's passionate about horticulture research and food security. In 2017, she worked with the University of Delaware on an agricultural sustainability project researching ways to decrease arsenic contamination in rice, which is a global phenomenon affecting the safety and security of our food supply. Upon winning the Delaware BioGENeius Challenge for her research, Preeti advanced as a finalist in the International Biotechnology Competition and presented her work to the bioscience executives at the Bio Competition in Boston last summer. Recently, she was also recognized as a 2019 Regeneron STS Scholar by the Society for Science. Through the course of her journey as a young agricultural researcher, Preeti has grown to realize the importance of plant science in addressing issues across disciplines and the impact that horticulture can make in global communities. Please welcome her now.
Thank you, Seth, for that wonderful introduction, and thank you everyone for gathering here today. I'm very excited to share my passion for agriculture and food security, as well as talk about the impact that we can make through horticultural research. So a little bit about me. I grew up gardening in my backyard with my parents, going to any botanical garden I can get to, and Longwood is probably the one I came to most. And my experiences with plant and soil really inspired me and made me into this happy panda right here, passionate about food security and food safety. So last summer, or in the summer of 2017, I got involved with a research project at the University of Delaware's Agricultural Department exploring arsenic contamination in rice. And I had the privilege of being able to present my work at the International BioGENeius Challenge in Boston, and I was honored as a Regeneron Science Challenge Search Scholar. During my time at Bio, I presented my rice research as the only finalist in the agricultural sustainability category seen up there. Something that was really shocking that I realized was that I didn't see any other young people speaking about agriculture. And it was not a hot topic anymore, and people of our generation seem to have forgotten about it. And that is a major problem. At this point in time, Americans can only identify 10% of the plants around them at any given moment. Today, most Americans don't understand how plants and biodiversity are important to human sustenance and are so intertwined with our lives. They don't seem to value these elements and the interdependence between the two. And this is particularly seen with the number of people who are leaving agricultural sciences versus the younger generation. So as you can see in this graphic, the number of people who are leaving agriculture, who are outgoing, is outnumbering the number of people who are coming in, the younger people. And it's very concerning because as our population as a whole grows, there's a growing unmet need for sustainable food supplies and plant-based medicines, and those are just not going to be found if we don't have people willing and able to find them. So a little bit about the symbiosis between plants and humans, because as I just said, it's not something that's very well understood by most people, and that's something that everyone in this room, I believe, can walk away from this presentation understanding and appreciating. So plants are very important, and if we all didn't believe this, we wouldn't be here today. But they're important in many different ways. They can be really useful and very important in going ahead with discoveries for new medicines, things that can cure human disease. And of course, they're very important for the human food supply. But it's not just the vegetables, it's the food that other animals eat. It's the food that other animals that we humans eat. And plants, for that reason, because they're so important to life, have grown with the cultures that have grown them. They've influenced many civilizations and cultures. And because of their importance in our world, they impact our climate, and they even breathe out the oxygen that we breathe in. And if that's not an example of symbiosis, I'm not sure what is. But last, and definitely not least, at the core of our planet's ecosystem are plants. And all of the biodiversity that we see around us wouldn't be possible without them. So with that being said, let me dive a little bit into my story and what my experiences were researching in this space. And I hope that through this I can strongly convince everyone in this room to go into this field and look a little bit more into the research opportunities available. So let me start with a problem, something for you to ponder. Arsenic contamination threatens both food security and human health across the world. 50% of the world's population relies on rice as a staple food. So as you can see, this is a very large issue, and I only started learning about it during my research with the Seyfferth lab at UD. So the Seyfferth lab itself is dedicated to investigating silicon soil amendments, which are things that you can add to soil that contain substantial amounts of silicon. And the lab was dedicated to investigating the effects of these silicon amendments on the uptake of arsenic in rice plants. So you may all be wondering, why are we looking at silicon exactly? So there are a couple of different reasons. The first being that silicon competes with arsenic in the soil to be taken up by the same root pathway in the rice plant. In other words, the same pathway in the roots that arsenic compounds basically sneak up into the plant through also take up silicon. And the second major reason is because silicon can change the state of matter of different soil nutrients and metals like iron. In fact, silicon oxidizes iron to produce a certain mineral called iron oxide mineral. And that's where I came in. I looked specifically at the effects of silicon on the iron oxide levels in the soil, because iron oxides bind arsenic, which means that if all of the arsenic is bound by this iron, there's so much less that the plant has available to take up. I did this by looking at the two different silicon materials that are bioavailable and natural products of the environment. Fresh rice husk, which is the protective covering on the rice grain, and its combustion byproduct, rice husk ash, which is considered agricultural waste in many of these rice-producing countries. And both of these materials are very rich in silicon. So between the two amendments that I studied, I found that rice husk ash was the most effective one at decreasing the bioavailability of arsenic uptake in rice, because it increased the presence of iron the most, which can then bind up more arsenic and prevent it from uptake. Something that was particularly unique about my experience researching at UD was that I got to research both indoors and outdoors. So I spent my summer mornings at UD's rice paddies collecting soil and pore water samples, helping to irrigate the paddies and monitor plant growth. And every day after I did my work outside, I would take in these samples indoors, and I would analyze them for different levels of iron and different nutrient contents. So one of the routine activities that I was involved in outdoors was something called gas fluxing, which is the process of tracking the fluctuations in climatic gases. And this is how it basically works. So the white cuboidal tent you see right over there, which is made of PVC pipes and tarp, is something that we placed on top of each of the paddies, which are the squares. And this would serve to trap the air in surrounding the rice paddies. And then we placed fans, which are attached to the stakes, as you can see over there, the black objects. And those fans allowed us to circulate that air in that environment and get a good reading of how the gases were fluctuating. And then we used gas sensors to collect data on the changes in gases such as methane and CO2. And all of this led up to our ability to analyze the greenhouse gases that could possibly affect our study outside, and to also maintain the atmospheric conditions at a constant level. Throughout the summer, I got to see the rice grow from seedlings, as they started off in June, as shown here, into full-fledged crops that were ready to be harvested. And I didn't only see this outdoors, but I also saw it inside the greenhouse as well. Rice is also a very important part of my own culture as a person of South Asian descent, and it's very important in my own daily lifestyle. So getting to see how the food I consume every day grows was very eye-opening and was a very gratifying experience. In the lab, I tested a variety of different things that I'd observed and collected outdoors. I measured the iron levels in pore water using techniques called spectrophotometry, as I'm seeing doing in the picture on the right. And I also got my hands a little bit dirty working with the soil to prepare it for different extraction procedures, where I was able to measure different iron levels. And so this was the result of my chemical extractions that I did on soil, to see how iron levels in the paddy soils were changing from the silicon that we added originally. And through my research, I was able to create an impact that was far-reaching than I'd expected before. I brought to the scientific community a cost-effective way of innovating to solve big problems like arsenic contamination. One that farmers around the world can readily access to implement as a practical technology on rice farms. Number two, I also helped to expand the horizons of food safety and sustainability by exploring ways to protect rice crops from environmental contaminants. And number three, I was engaged in and I also encouraged a multidisciplinary approach to problem solving in the field of agriculture, as I was using chemistry inside of the lab and I was using environmental science when I was measuring gases outside. So all of this leads up to the fascinating potential that agriculture has to make a true difference in the world. With the outdoors functioning as our experimental laboratories, the possibilities are simply endless. Through agricultural research, we have a pivotal role to play in tackling the world's most pressing crises, from malnutrition to hunger. And in our journey, we will be able to rebuild our historical connection with plants, gain global experience, and nourish our own awareness of the natural world. So every one of you in this room has the opportunity to make that impact by actively engaging in horticulture and agriculture. You are the future generation of change makers in the field, and you will be the ones determining the course that big issues like arsenic contamination and food scarcity will take 10 years down the future or longer. So I encourage you to start engaging in horticultural research to explore problems and find solutions to them. Exploring the nature around you can even help you connect better with yourself. So thank you so much for listening to my presentation, and I'm happy to open the floor up to questions. Yeah, so arsenic, well, let me start with where rice is mostly grown. A lot of countries in Asia, like Bangladesh, are major rice producing countries. And in these countries, the water that's used to irrigate these paddies is contaminated with arsenic that usually comes from pipes or is even a result of pesticide runoff. So this is a systemic issue that occurs from water quality. Any? Yeah, so there are scientists, especially across America, who are researching silica amendments in soil, and they're working with agricultural communities and institutions in countries like the Philippines and Bangladesh to share these solutions and spread the education about it to local farmers and local communities that know how the real operational process works. So in that, there are definitely more efforts that we can make as a community to bring scientists and farmers together to spread the impact of this. But yeah, that's something that people are starting to work on now.
In the silicone intake into the rice with the arsenic, so you're seeing it slows down the intake of the arsenic because it actually absorbs at the same rate as the water does from the plant?
Yeah, so it's not exactly about rate. It's more about when you have arsenic in soil and you add more of anything into that environment, you decrease the concentration of arsenic overall. So there's less of a chance that arsenic can be taken up when you have more silicon that can go through the same pathway. And silicon, in fact, when it goes up into the plant, has beneficial effects for the plant's immune system. So it can actually fight off disease like rice blasts. And that's another project that one of my colleagues in the lab is working on right now.
What are some of the economic challenges that are keeping some of these countries from changing this? Is it education or is it finances?
I think most of the challenge lies in organization and education. Kind of organizing the scientific community and encouraging people from science to take their solutions and impact it, working with people in farms. So there's still that divide between agrarian communities who have less education, who are from a lower socioeconomic status in developing countries, and people who are more technologically oriented. So I think part of the next steps in realizing the benefits of this is kind of bridging that divide. All right, thank you so much.